Hello, everyone, and welcome to today's webinar, Establishing a Robust 37-Marker Mass Cytometry Assay for Deep Single-Cell Immune Profiling of Whole Blood. I am Michelle Ashton of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Miltenyi Biotech. To learn more about our sponsor, please visit www.miltenyibiotech.com. Let's get started. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. If you have trouble seeing or hearing the presentation, click on the Support tab found at the top right of the presentation window, or report your problem by clicking on the Ask a Question box located on the far left of your screen. This presentation is educational and thus offers continuing education credits. Please click on the Continuing Education Credits tab located at the top right of the presentation window and follow the process to obtain your credits. I now present today's speakers, Mona Al-Maarri, PhD, Product Manager of Flow Cytometry at Miltenyi Biotech, and Adeeb Rahman, PhD, the Director of Technology Development at the Human Immune Monitoring Center at Icahn School of Medicine at Mount Sinai, New York. For a complete biography on our speakers, please visit the Biography tab at the top of your screen. Mona and Adeeb will now begin their presentations.
So thank you for the nice introduction, Michelle, and thank you all for joining this webinar. I will give you a brief background about what mass cytometry is exactly, and then I will talk about the advantages of this method. In the next point, I will tell you more about our off-the-shelf portfolio. Then I will introduce the technology of recombinant antibodies and the advantages for mass cytometry. And in the end, I will show you exemplary customer data. I guess most of you who are listening are familiar with mass cytometry, so I will keep it short here. In mass cytometry, cells are labeled with heavy metal conjugated antibodies similar to flow cytometry. Cells are then nebulized into droplets, and then they are vaporized, atomized, and ionized. This ion cloud that you can see between C and D is then filtered to enrich the heavy metal ions before they are quantified by time-of-flight mass spectrometry. The mass of the metals will then identify the expression of specific markers. Because the cells are labeled similarly to flow cytometry, this technique is basically a fusion between flow cytometry and mass spectrometry. When comparing both methods with each other, flow cytometry is very fast. The cells are not destroyed, which also allows for downstream experiments and sorting, and the handling is not as advanced as mass cytometry. Advantages of mass cytometry, on the other hand, are that you have almost no spectral overlap, therefore you don't have to compensate, and the panel design is much easier. Another point is that you don't have cellular autofluorescence because heavy metals do not occur in living cells. The results are on a single-cell level and at the same time allow for multi-parameter analysis because up to 40 parameters can be measured in one run. And basically, the same antibodies as for flow can be used. And because most of the antibodies available from Miltenyi Biotech were developed especially for flow cytometry, our antibodies are also suitable for mass cytometry experiments. And this is why we are actually providing pure antibodies for mass cytometry. Right now, we have around about 130 antibodies in the correct format available. Some of them are traditional monoclonal antibodies derived from hybridoma clones, and some are recombinant antibodies. But we are more and more switching to recombinant antibodies, which I will explain later on. They are provided in the format of 100 micrograms and 100 microliters, and in PBS only without any additives, because this format is necessary for the commercially available conjugation kit, because any additives could interfere with the conjugation process. If you would like to purchase a clone not available in the mass cytometry suitable format, you can easily request this antibody via our custom antibody design service when entering our webpage, since we have almost 1,000 different recombinant clones available, plus, of course, additional hybridoma clones. And the recombinant clones all have REA in their clone name so that you can identify them easily. Now in the next part, I will explain more in detail why it makes sense to switch from polyclonal or monoclonal antibodies to recombinant antibodies. And in the past years, there's a growing concern in the scientific community that many published results are not reproducible by other researchers, as reflected by multiple publications and articles. Antibodies are considered as one of the key variables in the system that add to irreproducible results. In 2015, various articles were published in Nature highlighting the importance of antibody validation, and they also called for a new generation of antibodies, which are the recombinant antibodies that are defined by their DNA sequence. In 2018, even more papers came out demonstrating that hybridoma-derived monoclonal antibodies are often not very specific and that recombinant antibodies are the way we should go. I will quickly explain you the reasons for this, and hybridoma clones, so the ones used for traditional monoclonal antibodies, are generated from a fusion product of antibody-producing B cells and immortal myeloma cells. Sometimes the hybridoma-generated antibody products also contain the contaminating light chains, which are shown here in green, which are produced by the myeloma cells. In an ideal situation, the same antibody is also produced over a long period of time in batches. However, when they are in culture over several passages, spontaneous mutations can lead to genetic drifts, and an antibody product different from the expected product will be the result in the end. In addition, when a hybridoma cell line dies out, then the antibody clone is practically lost forever. The antibodies can also be contaminated by the immunoglobulin coming from the serum used for cultivation, which is shown here in gray. Such contaminations decrease the purity of the product, and furthermore, the impurities can vary from lot to lot, making the hybridoma-derived product inconsistent. Recombinant reaffinity antibodies, on the other hand, are genetically engineered. The defined DNA is introduced into the mammalian cells, which are programmed to only express the product of this DNA sequence. Because they are not derived from B cells and the cells are cultivated in defined culture conditions, no contaminating light chains or antibodies can be present. And furthermore, the cells are reproducible when the cell line might die out. Due to all these aspects, lot-to-lot consistency as well as purity of the product is ensured. And because they are all genetically engineered, they can also be altered in a defined way. So in this slide, I will show you the features of reaffinity recombinant antibodies and why this is actually beneficial for mass cytometry experiments. They are recombinantly generated, leading to lot-to-lot consistency and highly reproducible results. Due to the expression in standardized mammalian culture systems, no unwanted Ig impurities occur. Furthermore, as I said before, because they are genetically engineered, they can be altered in a defined way. Therefore, we mutated the Fc region so that no Fc blocking is necessary and background signal is reduced. They also have a human IgG1 backbone, and because of this, no isotype-dependent variation during metal conjugation can occur. And all of this is not only theory, because Miltenyi already decided very early to count on recombinant antibodies. So our first reaffinity antibodies have been released in 2012. In 2015, the most common markers were available already as recombinant versions, and now in 2019, we have the largest portfolio of roughly 1,000 recombinant clones that are optimized for flow cytometry. To also address the reproducibility crisis even more, we will extend the communication on how we validate our antibodies to you as a customer. And before I hand over to Adeeb, I will quickly show you some customer data. Due to the high purity of recombinant antibodies, the comparison of flow and mass cytometry data using the same clone becomes highly reliable. And here's one example showing data from Carl Johann Malberg. He used the same reaffinity antibody for the same reaffinity clone for flow and mass cytometry, and as you can see, the results using two different methods are very consistent, showing the same population size. Another example from our customer Amir Horowitz shows that the separation of the positive and negative population is nicely visible. Below you can see a t-SNE plot, and in a t-SNE plot, as many of you might know, many parameters are taken into account and therefore impurities coming from the product might already mess up your results. I chose this example because it is known that NKG2A and NKG2C are mutually exclusively expressed. And as you can see, the results show nicely the expected expression pattern because the one marker is only visible in the left panel and the other one is only positive in the right panel. As a summary slide, I collected the key characteristics again, and most of the antibodies for mass cytometry are recombinantly generated in a defined expression system, which of course leads to a high lot-to-lot consistency, as explained earlier. They all have the same isotype, making metal conjugation very consistent. They are validated for flow and are therefore suitable for mass cytometry. Furthermore, we offer more than 1,000 recombinant clones, which gives you a high flexibility to choose your panels. And last but not least, they have a mutated FC region, reducing the background signal. And with this, I would like to hand over to Adeeb Rahman.
Thanks, Mona. Following on Mona's presentation, I was going to go through and show you an example of a specific project in which we've been deploying some of these REA antibodies for immune monitoring purposes. But before getting into that, I want to provide a little bit of context for where this work is being done and kind of how it falls into our broader range of operations. So the Human Immune Monitoring Center at Mount Sinai is a center that aims to combine a range of technology platforms together with computational, technical, and scientific expertise and bring this to bear on a range of patient cohorts to derive biological insights, particularly in the setting of trials. And one of the challenges in doing this sort of work is really being able to balance sort of innovative assay development with rigorous standardization that's required to be able to minimize kind of the experimental and technical variability that can often compromise data in this sort of setting. And so our general mode of operation is to acquire blood or tissues from patients, and then we do have a large sort of arsenal of both technologies as well as basic sample collection processes that we can bring to bear on these tissues with the goal of ultimately generating a range of data types that can hopefully help to inform disease mechanisms, identify biomarkers, and potential biotherapeutic targets. And for the purpose of today's discussion, I'm really only going to be focusing on mass cytometry or CyTOF, which Mona already introduced, and we find a very useful technique for performing these sort of high-dimensional single-cell analyses. And so the specific study I'm going to be talking to you about today is part of a broader study that's titled Activate, the goal of which is really to evaluate the effect of the vaginal microbiome on the developing immune response of infants. And so the hypothesis here is that exposure to the vaginal microbiome has an impact on the development of pediatric food allergies. And so this is a clinical trial being performed at Mount Sinai. And there's 3 arms of the trial. There is one cohort of conventional vaginal delivery, a second cohort of C-section deliveries, and a third cohort of C-section deliveries who are seeded with vaginal microbiota. The infants in each of these 3 arms are being followed for 12 months. And the primary goal of the study is really to evaluate the impact of this on the incidence of food allergies. But in parallel, we have sort of a second research arm, which is to also conduct longitudinal profiling of immune cells during this 12-month period to be able to see if we can, determine correlates, immune correlates of these different sort of delivery modes as well as exposure to different microbiota, and in addition to see if we can find immune correlates of development of pediatric allergy. And so this kind of study has a few specific sort of challenges associated with it. One is that this does deal with neonatal samples, and so the amounts of blood that we can collect at any given time point are quite small, typically under a milliliter. There's also a challenge that it's a fairly large study in scale. So each of those 3 arms has a pretty large number of patients. There's several longitudinal time points, and this results in over 500 samples that are going to be collected and processed over several years. In addition, the goal is for this to be a multi-site study with several clinics contributing samples, and this presents additional complications for having a streamlined workflow that can't necessarily be deployed easily at each of those clinical sites. And so what I'm going to talk to you about today is sort of the process of how we went about developing a workflow for the purpose of the study. And I'm going to cover sort of many of the kind of key best practices, I think, that are relevant in any sort of study of this kind of magnitude, starting with sort of the target selection, antibody selection, how we go about conjugating and validating a panel, and then some considerations in actually developing a streamlined experimental workflow to be able to facilitate data analysis down the line. And one spoiler alert is that the study is just getting underway, so I'm really talking about the development of the methodologies for the study. I'm not going to be telling you anything about the outcomes of this research study, so sorry to disappoint there. All right, so in developing any sort of mass cytometry study, clearly your insights in any of these sorts of projects are largely driven by your antibody panel that you develop. And so developing an effective antibody panel is one of the kind of primary challenges and arts, I think, of ultimately gaining useful insights from a mass cytometry study. And generally at the HIMC, we take sort of a three-pronged approach to developing an effective panel. We have a set of markers that are required to comprehensively identify major cell lineages. We provide sort of a second tier of markers that help to provide deeper stratification of specific lineages that are relevant in the context of a given study. And then lastly, we try and identify markers that are potentially going to vary dynamically in the context of that study. And in developing this panel, or in sort of balancing these three arms across multiple studies, there's always this challenge of being able to kind of optimally customize a panel to gain, maximize your study-specific insights, and being able to balance this with the need to be able to standardize at least a subset of markers to facilitate cross-study comparisons and insights. And in doing this, kind of that orange category at the top, the markers to comprehensively identify the major lineages, is one of the easiest to tackle because there's kind of a fairly broad range of literature supporting what those should be. The other two really do require sort of more understanding of the context of the specific study and the nuances in that context. And so for this particular study, that first set of markers, this is sort of a list of about 13 antibodies that we sort of use as our core set of markers that we think is the minimal number of markers needed to identify most major circulating immune cell subsets. And kind of with this, we can do a pretty good job of deriving that kind of broader heterogeneity of the circulating immune system. We supplement this with an additional set of markers to provide sort of deeper stratification of specific cell types that we think are relevant for this specific study. For example, things like CD25 and CD127 to help us identify Tregs. We have a few sort of allergy-focused markers there, things like Fc-epsilon receptor and CRTH2. And then we also had a hypothesis around the study involved in homing, and so there's a pretty big range of kind of chemokine receptors there, which are also used in some context to help define polarized T-cell subsets. And then we had this final set of markers that we think also provide some additional subset stratification, but also expect are going to potentially vary dynamically in the context of inflammatory immune responses. And so having kind of put together this list of potential targets that we think are going to be informative in this context, the next step is identifying antibodies against this target. And there's sort of several considerations in picking antibodies. Obviously the specificity and the sensitivity of individual clones is a big one. And as Mona has already described in her section previously, the recombinant antibodies provide a range of advantages that we wanted to take use of in the context of this study. In particular, one of the points that Mona didn't touch on, which I think is relevant for mass cytometry, is the fact that all these antibodies have a constant Fc region. Presumably means that when you are custom conjugating antibodies, you can get much more uniform conjugation of different antibodies because you don't have any sort of variation in the available residues for conjugation in that Fc region. And kind of prior to starting this project, we had already generated some data showing that the performance with the REAs was at least comparable, if not better, to our experience with several other antibodies targeting those same clones. And so we went ahead and worked to develop a largely REA-based panel for these antibodies. And I should note that when we first started this project, which was I think a couple of years ago, most of the antibodies were in fact not available as off-the-shelf antibodies from Miltenyi, and so this did require a request to custom formulate almost all of these to make them compatible for mass cytometry, essentially to have them in a stabilizer-free format. Since then, however, almost all of these are available as catalog antibodies, which I think would make this an easier workflow for folks looking to do this down the line. And so the last component here then is we've got our target selected, we've got our antibodies. The next is to actually conjugate these to isotopes for mass cytometry. And there's sort of two primary considerations that we take into consideration when deciding what isotopes should be assigned to which antibody. The first is sort of the relative transmission efficiency on the instrument, also known as the mass response curve. So mass cytometers do sort of have an optimal detection range, which is typically in the range of about 150 to 172 or so, as you can see from this publication from a couple of years ago. It is somewhat instrument specific, but what this means is that if we expect that particular antigens are going to be present at relatively low abundance, to maximize detection of those antigens, we want to couple them to isotopes that are optimally present in this sort of high efficiency part of the mass range, whereas relatively high abundance targets can be relegated to sort of the left and right side here, the upper and lower masses, and where we can still expect to see decent resolution of those populations despite the lower transmission efficiency. A second equally important aspect is the consideration of signal interferences. So mass cytometry has, in general, much lower spillover than flow cytometry, than essentially fluorochromes. However, that doesn't mean that it's non-existent, and there's essentially three primary sources of the signal interference, which have been described in a few publications. I've got one of them cited here. One of the ones is essentially isotopic impurity. So when we have an isotope, like for example a neodymium isotope here, with an expected isotopic mass of 145, if that isotope isn't entirely pure, it could be contaminated with some percentage of 146, for example, and that would be one source of apparent signal interference or spillover. A second source is oxidation, where any individual isotope can end up being coupled with an oxygen, resulting in sort of a plus 16 signal interference. The third category is actually just abundance sensitivity, which reflects the fact that high detection of an isotope may have a little bit of detection in the adjacent channels, which is relatively minor compared to the other two sources. And so, taking all of these points into consideration, we went ahead and I assigned isotopes for each of the individual antibodies in this channel, and then we went ahead and custom conjugated all of these in-house using MACS X8 kits from Fluidigm. So once these antibodies have been conjugated, the next step is validating that the conjugation has actually been successful. And there's a few ways of doing this, but our preferred method is to actually use antibody comp beads. So I'm sure most people in the audience are probably familiar with these. These are essentially beads that are coated with capture antibodies, and you can essentially take these beads, mix them with a given antibody, the beads end up getting coated, and then you can run these on a flow cytometer. And these are often used for performing, essentially generating compensation controls by conventional flow cytometry. And they're also quite helpful to use in mass cytometry. Two sort of considerations in doing this, though. One is that in mass cytometry, we don't have traditional forward or side scatter measurements, and so these beads, in the absence of bound antibody, are essentially invisible to the instrument. They don't have any sort of native signal on them, and that can create some complications in terms of detection of the beads. So it's helpful to have some way of labeling the beads to ensure that they are detectable. One of the ways that it turns out you can do this is just to incubate these with the iridium intercalators that we use for conventional mass cytometry cell detection. And while these intercalators are supposed to bind to nucleic acids, they do seem to have sufficient, non-specific binding to these beads that you can see that kind of blue population there that has sort of enough IR signal to be detectable. A second consideration here is what beads to use. So traditionally, in most of our workflows, we were using the ultracomp beads developed by eBioscience, but the REA antibodies in particular, presumably because of their mutated human Fc region, don't actually bind to those beads. So for this particular panel, we were largely using the MACS Comp REA beads from Miltenyi. And I should also note there's a relevant reference here from Lisa Budzinski out of Hendrik May's group, who've shown a similar workflow using osmium to label these beads, which is another way of facilitating their detection by mass cytometry. So having generated these beads, you can see that they work very well for being able to QC that an antibody has actually been conjugated specifically to the isotope we hope it has been conjugated to. So you can see the CD19 here is giving us a nice clean signal in 142, as expected based on the metal it was conjugated to, and likewise for CD66B, CD14, CD38. Another useful aspect of this, as I touched on a few slides back, is that mass cytometry does suffer from certain aspects of signal spillover. And these beads offer a nice way of being able to visualize that. So here you can see another example of the 145 neodymium, which is the primary channel. But you can see that there is detection also of the signal of that CD4-145 antibody in the 146 channel. And that reflects the 3.9% known isotopic impurity of 146 in the 145 neodymium. Similarly, we can see that oxide at the plus 16, 161 mass, that in this case was calculated to be about 1.5%. And if we do this for the entire panel, it actually helps to recapitulate this sort of spillover chart, which many users of mass cytometry may have been familiar with already, where you can see that for every antibody, we can detect the relative amount of spillover in any of the channels due to either oxidation or isotopic impurity. And a method that's recently been developed is to essentially use this to actually compensate CyTOF data to be able to correct for this. Though, admittedly, this is something I don't spend too much effort doing, partly because we don't find the compensation really helps to correct for the underlying issues of mass spreading or sort of signal or dilution of signal resolution, but based on expansion of the negative populations with compensation. All right, so that's essentially the first step of validating these antibodies with beads to ensure that we've conjugated the correct mass, we're seeing signal where we expect to see them. The next step then is to do a second round of validation with actual biological samples and validation and titration. So here we tend to use sort of a three-step approach. We start off with the antibodies that are essentially four canonical subsets that show kind of very clear bimodal distributions, and these can typically be titrated in the absence of any sort of cellular context. So simply titrating CD3 on an otherwise unstained sample still allows you to resolve your CD3 positive and negative populations fairly well and determine what is an optimal concentration to stain at. And I should note with mass cytometry, we're often not using fully saturating concentrations of these antibodies. Instead, we're looking for optimal separation of a positive and negative population. And one of the metrics we use for this is the average overlap frequency, which is a method we published a couple of years ago that essentially calculates how much separation you have between a defined positive and a defined negative, and we try and maximize this metric with our titrations. So the second set of titrations is antibodies for which some cellular context is helpful. So for example, when titrating an antibody like CD25, it's helpful to be able to specifically gate on CD4 T cells, as you can see here. And then within those CD4 T cells, we can now evaluate CD25 and CD127 and find the concentration that allows us to resolve that Treg population. And again, in the absence of having this cellular context, if we were just to be titrating CD25 on the bulk population, it's more difficult to evaluate what is a known positive and known negative for this sort of exercise. And similarly, here's another example of CXCR5 and CD45RA below. So this is also actually useful for antibodies where that are sort of expressed in a heterogeneous format. So one example here is CD16. This is being titrated in whole blood, and you can see that B cells here are serving as our negative control, neutrophils are serving as our positive control, and sort of eyeballing this with the neutrophils, it looks like we're getting close to saturation of this antibody at about 0.5 nanograms per microliter of whole blood. However, CD16 is also expressed by other cell types in blood, and so by being able to define that cellular context, we can actually see now that even though 0.5 or even 0.25 nanograms per microliter is sufficient to resolve our CD16 positive neutrophils, it looks like we need much higher concentrations to be able to resolve our CD16 high NK cells and our CD16 high monocytes. So it's another context where titrating an antibody with sort of known cellular context is helpful in determining optimal concentrations. And then the third and final category of antibodies are those that are essentially dynamic activation markers and may not be expressed at sufficiently high expression levels in sort of a normal healthy donor sample. So an example here is PD-L1. We're using sort of CD8 T cells as a negative control, CD14 monocytes as a positive population where we potentially expect to see some expression. And we can see in general there's relatively low expression here. It does seem like we're getting a dose-dependent increase in PD-L1 expression on monocytes. But when we look at our CD8 T cells, which we expect to be predominantly negative for PD-L1, we can see a similar dose-dependent increase here, suggesting that this is largely an increase in background. And so based on this, one might conclude that our PD-L1 conjugation has failed. We aren't getting effective detection. But of course, the alternative is that we simply aren't seeing sufficient expression of this. And so now we can repeat this experiment, but now using whole blood spiked with some 48-hour PHA-stimulated PBMCs, where the interferon is expected to drive upregulation of PD-L1. And now using this sample, we can kind of gate on both the healthy adult blood sample as well as those PBMCs that have been spiked into it. And we can see that, again, while we don't see much expression on the healthy old blood, we can see very robust expression on those PHA-stimulated monocytes. So we know that our antibody's working. We know our conjugation succeeded. This just tells us that the physiological dynamic range of this in a healthy blood sample is too low or below the level of detection. But hopefully in a perturbed setting, expression might increase to the point that we can, in fact, resolve this. So once we've sort of validated each of these antibodies individually and in sort of their appropriate context, we can then go ahead and test the entire antibody panel. to confirm that all the antibodies are being expressed sort of on populations that we expect them to, and to see how those populations may resolve. So I'm just using a t-SNE analysis here that I ran in Cytobank. I'm just showing you representative data from 12 of the markers in the panel, some of the kind of key canonical ones, and again, there's several more antibodies here. But broadly speaking, you can see that sort of you have this large population of neutrophils in the center, characterized by CD6xB and CD16. There's some eosinophils on the side there, there's 6b and 16 negative, and so on and so forth. And another way to be able to evaluate this sort of data is to actually perform some clustering. And one way in which we do this is to actually use flow sum clustering and cluster annotation using the Astrolabe diagnostics platform. And so here you can see on the left that there's a heat map where every column corresponds to one of the markers in our panel. Every row is a cluster defined by expression patterns across those. It's a little bit hard to read the text there. And we can also go and take those clusters and actually map them back onto our t-SNE plot and kind of further confirm how all of these relate to each other. I'm not going to belabor this, but essentially a deeper dive into this suggests that all of the antibodies in our panel are in fact being expressed on the cell types we would hope they would, and they're providing sort of interesting resolution of that biological heterogeneity. So having sort of optimized this panel, our next step was we wanted to just confirm how much blood we actually need to be able to perform this study. As I mentioned at the start, this is a study with pediatric and neonatal samples, so we really want to be able to use as little blood as possible. And so we essentially titrated down these antibodies with given blood volumes, and we're able to see that at least 4 major populations, what is the lowest concentration or the lowest volume of blood at which we're able to see sort of stable frequencies of these. And we, from this analysis, ultimately concluded that 200 to 250 microliters is sufficient for resolution of most circulating immune populations. There's obviously greater heterogeneity in some of the very rare DC subsets and things of that sort, but it seemed like it would be doable in the context of this particular study. So the last aspect I want to touch on here then is how to actually go about implementing this panel in the setting of kind of a multi-site sample collection with these minimal samples, as I touched on before. So one way in which we often deploy CyTOF is to do sort of a standard blood collection. We isolate PBMCs, we cryopreserve PBMCs, and then when we're ready for analysis, we thaw those samples in batches, and we perform our staining. So this is kind of a fairly effective workflow. The primary downsides are that it's quite labor intensive because it does require that PBMC isolation, which also requires relatively larger blood volumes for effective isolation as well as cryopreservation and storage. And it can also be somewhat prone to technical artifacts, though being able to batch samples for analysis does help to mitigate that to some extent. But in this case, that really isn't feasible, partly because of the blood volumes we're dealing with. A second approach is to simply use whole blood monitoring and to essentially collect blood, ship that blood to a central lab, and perform real-time staining and acquisition. So this is labor-intensive largely because it requires that real-time staining and processing. So if it does, if one needs to assemble this panel on a regular basis, that's also somewhat error-prone and prone to technical batch effects. And then one of the challenges also with collecting blood at multiple sites and shipping them to a central site for analysis is that you have to have very strict limits on the time points, because obviously as blood is sitting in a tube, you would expect that there are going to be some phenotypic changes to these populations that might be happening spontaneously that are going to provide technical impacts on ultimate data quality. So this didn't seem optimal either in our workflow. So a third option is to collect that blood and to stabilize it at the site of collection. And one buffer that we use routinely for this is the Smart Tube buffer, which is an effective way of being able to fix, stabilize, and ultimately cryopreserve blood. And this cryopreserved blood can then be, as it's now essentially frozen, can be shipped to a central lab for batch staining. So the advantage of this is it's a very efficient workflow because stabilizing blood is much easier than staining blood. It's essentially just the addition of a single buffer to a tube and then shifting that tube to a freezer. A downside, though, is that because fixation is involved, there are certain epitopes that are going to be impacted by this. So I'm showing 2 examples here. So you can see that CRTH2, which stains basophils very effectively in whole blood, that staining is completely lost looking in that Smart Tube fixed whole blood. Another example here is CXCR3, chemokine receptor, where you can see that we have sort of a different problem where, in the whole blood, we can see kind of that nice resolution of CXCR3 positive versus negative CD4 T cells. Monocytes are predominantly negative. But in the Smart Tube fixed whole blood, we sort of lose that positive discrimination on the CD4 T cells, and we gain this non-specific kind of background staining on the CD14 monocytes. So certain marker distributions are definitely affected by fixation. And if you're curious, this wasn't done with the REA antibodies, but we did recently publish a comprehensive screen of 300 plus antibodies that we evaluated for fixation compatibility using sort of similar methods to what I've shown here. So the challenge there with option 3 is that it's an efficient workflow, but it's not compatible with all the markers in the panel. So ultimately, our preferred workflow is really to collect the blood, perform real-time staining on site, so the blood essentially is stained fresh, and then it is fixed and stabilized using the SMART2 buffer, and then shipped to our central lab for ultimate data acquisition. But in order for this workflow to work, the real-time staining has to be as sort of easy and simple as possible to be done at these sort of remote clinics that don't have much experience with mass cytometry or any sort of biospecimen processing. And so the way we accomplished this was to lyophilize our entire panel and to dispense this panel in single-use cryovials. So what this essentially looks like is we have these cryovials that have a little a lyosphere of antibody in the vial. I should mention that we did this commercially with a company called BioLife. And so what can be done at the clinic is that we simply take 200 microliters of whole blood, we add it to the antibody lyosphere, we incubate that for 20 minutes, and then we add our SMART tube stabilization buffer and transfer this tube to the freezer. And then those frozen samples are shipped to our central lab for the final and DNA intercalation as well as data acquisition on the instrument. And just to show you that this workflow seems to work quite well, so I'm showing you up top the same panel in its liquid versus lyophilized form. And here's just a range of antibodies, and you can see that generally speaking, we're seeing very good preservation of these staining profiles in both the liquid as well as the lyophilized antibody. These are some primary canonical immune staining markers. We're seeing the same thing here for some of the chemokine receptors in the panel that show these kind of slightly more subtle expression patterns, all of which seem to be preserved quite well. Another way to evaluate this for the entire panel, or rather another advantage of the lyophilized panel beyond simply looking comparable to the liquid panel is that we expect that this does provide enhanced stability of our panel over long periods of time. And one way to evaluate this is to do an accelerated stress test at elevated temperatures. So you can see up top we have our lyophilized panel, and it's being either stained fresh or it's been stored at 45 degrees Celsius for up to 8 weeks. And you can see that for that entire 8-week period, we're getting broad preservation of the antibody staining profiles that we get despite stressing this panel to a pretty significant extent. And if you contrast that with a conventional liquid panel at the bottom, you can see that even a single week at 45 degrees pretty much means that most of the antibodies in the panel are toast, which is not entirely surprising. Another way of evaluating that is using that average overlap frequency metric I mentioned earlier. You can kind of see in the little heat map there that single week has really compromised expression of most of the markers in the panel, whereas it's being preserved quite well in the case of the lyophilized panel. And so this gives us confidence that we can actually ship these things out to people, at ambient temperature. They can sit in the clinic for however long is needed to be able to perform the staining, and then we can expect to get fairly robust data back to us. To also evaluate this workflow actually works, we did a bunch of testing just to show that staining replicate blood samples with these panels is reproducible. So here's one example from one donor where we have 3 replicate aliquots of 200 microliters of blood from the same tube that have been stained separately with in independent cryovials. And I'm just broadly characterizing some of the major immune populations here, and you can see that essentially our frequencies of these subsets are highly reproducible over time, and the t-SNE plots kind of show you that the general staining expression is also highly reproducible across these aliquots. And calculating this across 5 donors, we find that our CVs are typically less than 5% for all defined immune subsets. So this is purely technical reproducibility. Another important consideration is also sort of biological stability of these populations. So this is a similar experiment, but here, instead of taking replicate aliquots from the same tube of blood, we had the same donors come back at 2-week or at 1-week intervals, and so we have a blood draw at day 0, day 7, and day 14. And similarly, again, you can see our broad expression profiles look quite similar, but we do see more variability here in the frequencies of some of these subsets. So, for example, the monocytes you can see there are elevated at day 14 relative to day 0. And this makes sense, but it does tell us that there is going to be some expected, just normal biological variability in some of these populations. And this helps us set kind of thresholds that we would expect to want to look for changes beyond in the case of our cohort. So essentially, we might be able to conclude from this that a 3% change in monocyte frequency might just be part of normal biological variation, whereas perhaps a change of 20% is going to exceed that normal biological variation and reflect something interesting that might be happening in that cohort. All right, so based on all of this validation data, we were fairly confident we could take this workflow and now deploy it in the setting of our clinical trial, and that's what's happened currently. So we have our various sites, all of which have been shipped these antibody lyospheres. And the goal here is now to, again, implement this workflow at various time points along that 12-month window of the first year of life. And eventually all those samples are going to be aggregated, shipped back to us, and we're going to perform mass cytometry on that. So hopefully in a couple of years, I'll be able to give you some updates on that. To conclude, I've walked you through some of the key steps at each of the some of the key considerations in developing and deploying a study of this sort, starting from sort of the target selection, the identification of clones, conjugating those antibodies, and validating and titrating them, and ultimately then lyophilizing that panel and sort of revalidating the lyophilized panel. And then, essentially these panels have now been distributed to our clinical sites. That real-time whole blood staining is happening currently, and ultimately those samples are hopefully going to be analyzed to identify correlates with pediatric allergy. Right, I'm going to end there by acknowledging the entire HIMC team, in particular the CyTOF team, Brian, Bhaskar, Jeff, and Daniel, and our collaborators Cecilia Barron and Shuda Agash, who've been running a lot of the allergy-specific validations around this. I also wanted to note that we're always hiring, so if this sounds interesting to you, let me know.
Thank you, Mona and Adeeb, for your presentations. A quick reminder for our audience on how to submit questions. Simply type them into the drop-down box located on the far left of your presentation window labeled Ask a Question and click on the Send button. Our speakers will answer as many questions as time permits. Okay, the first question is, are there any downsides to antibody panel lyophilization?
Sure. I guess there's two primary downsides. One is cost. So it isn't cheap, at least to lyophilize these things commercially. It does scale very well with volume. So this wouldn't really make sense to do in a study where you only have sort of 50 or so antibodies or 50 or so vials that you need to lyophilize. But when doing sort of several hundred or thousands of tests, this does become fairly more cost effective. The other downside is that not all antibodies do tolerate lyophilization. So I think we did get lucky in the context of this panel that everything seemed to work out quite well. But we have had prior panels where at least one or two antibodies in the panel have not tolerated lyophilization, need to be spiked in. So that is a potential concern. So I think the key is to perform sufficient validation of the panel pre and post lyophilization to be confident that it's working well. Once the panel has been lyophilized, then I think things are pretty good. The one other caveat there is that there is a tendency for some of these lyophilized antibodies to form some antibody aggregates, and this kind of manifests as kind of a diagonal streak in mass cytometry data. Filtering the panel through a 0.1 micron filter, which is kind of our standard workflow for CyTOF panels in general, does a very effective job of mitigating this. In the context of this particular study, just because the whole blood is being added directly to the tube, we expect that this is likely going to be an issue. But it can generally be taken care of at the data analysis step.
Great, thank you. All right, your next question is, are there other ways to store antibody panels, for example, freezing?
Absolutely, yeah. So this is a nice technique that was, I think, published again by Hendrik Mai's group, essentially taking an antibody panel and sticking it at -80 degrees does a very effective job of preserving many of these antibodies for long-term storage. I should note that conventional 4-degree storage does not work well for mass cytometry panels. We see all sorts of strange data artifacts that happen when a panel sits around. But freezing it does seem to be quite effective, and I think is a more accessible workflow for many labs that don't want to embark on lyophilization. I should note the one disadvantage of that is that shipping frozen panels and using them in a multi-site setting is obviously much more complicated. So for this particular workflow, I don't think that's as effective, but certainly for single-site studies where samples are simply being stained longitudinally over time, frozen panels are an effective strategy, I think.
Thank you. Why don't you offer antibodies directly coupled to isotopes?
This is pretty simple, and this is just because we don't own the technology, so we also don't have instruments to test antibodies that would be directly coupled to isotopes. But because so many customers always asked for the antibodies via our custom antibody design service, we decided to make an off-the-shelf portfolio out of it, so to offer the pure antibodies, but we cannot offer them coupled to isotopes.
Thank you. It looks like we have time for one more question. What is the minimum amount to order an antibody via your custom service?
So usually via our custom service, you have to order at least 500 micrograms because, yeah, this is just amount where for us it makes sense to produce it for the customer. But for CyTOF, we decided to go with 200 micrograms because we know that the conjugation kits are usually made for 100 micrograms, and 200 microgram is the minimum we can afford for the customer. So this is, yeah, kind of a service for the CyTOF users. So this is only possible if it's requested for a CyTOF antibody, not for the other antibodies.
Thank you. I would like to once again thank Mona and Adeeb for their presentation. Do you have any final comments for our audience?
None for me.
No.
Thank you. I'd like to thank the audience for joining us today and for their interesting questions. We would like to thank our sponsor, Miltenyi Biotech, for underwriting today's educational webcast. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's event. Until next time, goodbye.